So our final speaker is Florian Berger from theoretical biophysics. He made a long way, I think, from theoretical physics to biology. So it's interdisciplinary in itself. And the title is Stochastic Dynamics and Biology, Thermodynamics to Control.
Yeah, thank you very much for giving me the opportunity to talk here. So on the way to the room today, I was thinking about, okay, when was the last time that I gave a talk to a more mathematical inclined audience? And I think it was 18 years ago, and I had to give a seminar about Young tableaus in group theory. But as a student, I was so afraid of that, so I had prepared 2 weeks for it, so it was perfect. But let's see how today goes. Will be also a little bit, I think, more entertaining because I thought it's the last talk, so I will also show some videos, what I think is incredible and remarkable about biological systems. So what we are doing is actually we try a physics approach to understand biology or to contribute to the understanding of biology. And then, of course, you can ask, you said, what is the physics approach? And I want to argue that physics was very successful in the last 400 years because it's based on 4 steps. So usually we take a system, we observe it, and then we have a mathematical description for it. But then we don't stop actually, because then I think the important point is that we also want to understand how the system behaves if we perturb it. And then if we have a complete description of the perturbation, then we can also make a very clear prediction how the system works, and then we can build actually things because we know how to control it. And here, for the example of a pendulum, you maybe know that all these skyscrapers, they have a very huge mass on top with dampers to actually damp out resonance vibrations that these buildings do not collapse. And I think this approach actually is very important or very successful because it automatically leads to a certain predictability of the system. It's highly reproducible because we base everything on numbers. And I also believe we often can find causalities in it. So this is maybe also why I think it's still important to do these approaches. Also, we will have probably soon some neural networks and they can just create simple differential equations from a bunch of data, right? But then we still don't understand a causal relationship. Just a side remark, I mean, because I work now with biologists, some biologists argue physics was so successful in the last 400 years because physicists only ask very simple questions, right? So because if you talk about life and difficult biological questions, then it's a different.
At least I can ask.
Exactly. I think maybe also why, yeah, it's how you define success, right? But so I think there are some problems why this approach is still not so successful in biology. And this is because in biology, we often have to deal with nonlinearities. And then by definition, I want to argue these are non-equilibrium systems. And for non-equilibrium systems, we unfortunately don't have the same tools to describe also physical systems. And then I also think that biological systems are actually poised on a very interesting level of complexity, because if a biological system would be like a gas, so it would be like a lot of particles, but they all behave kind of the same, then we can just use statistical mechanics to describe it. But biological systems, they have something sometimes also called organized complexity, where you have still not a lot of degrees of freedom, but you know what are the relevant degrees of freedom, but they could be all kinds of different. So it's very difficult to reduce also to a set of variables that you want to study. And then, of course, you have stochasticity and variability in these systems, although the function is usually very well conserved. So where are we now? So where's our group now? So for the last years, we were mostly working here. So we are observed and we described with mathematical description, and sometimes you can make good prediction of these models. And then now what I want to do also in the next years is to push forward more into this direction. I want to tell you today a little bit about what we can learn from perturbations, and I will show you some things about control. However, for biological systems, what I think is very relevant is actually the point of non-equilibrium, because I think this was also actually pointed out by Erwin Schrödinger in the 40s, and he gave a very interesting lecture about biological systems. So he's said in this that living matter evades the decay to equilibrium. For me personally, I think this is probably the most fundamental aspect, how you can also distinguish living systems from non-living systems. This has been also lately picked up again by Paul Nurse. He wrote this small book about what is life, and there's also what is life, several books. So maybe I should write one when I retire.
You want to have answers?
I don't have an answer, let's see. But yeah.
This is something for retired people, that's what you say.
Yeah, I still have 20 minutes.
That's your Nobel Prize, though. First you have to get the.
Yeah, I think I'm too old for that one. But what I want to argue is actually that if they are somehow escaping to decay to equilibrium, so then they also must dissipate something in the environment. So it's a non-equilibrium system. What I want to discuss today is, can we actually also quantify this dissipation in these systems? So one problem with this is now I use terms, from equilibrium. I talk about equilibrium. So this is thermodynamics. Also, what is heat? Can we define heat in a non-equilibrium system or in a stochastic system? And I think here I just want to show you next video, because I want to make the point that these systems are highly stochastic. So what you see here is a neuron. So that's recorded in the cloud building. And these are transport of material within the neuron. So it's a super stochastic system, and there are a lot of things going on in these cells. However, I'm lucky, because in the last 15 years, there's also a new field evolving in theoretical physics, which is called stochastic thermodynamics. And the idea of stochastic thermodynamics, very briefly, if I summarize it, so in a classical thermodynamical way system, we can define two states, for example, for a gas, and then we can drive the gas between the two states, and then we can calculate the free energies. However, we know that thermodynamics or macroscopic thermodynamics dictates that we will always lose here energy, or we have to dissipate work into the environment. So that's the second law of thermodynamics. So the problem is now when you go to small systems like molecules, they are surrounded in a thermal environment, then this actually breaks down apparently. So the problem is here that, for example, if you have a molecule, so this is depicted here, and what you can do with optical trap, this is nothing else than a pico-Newton machine. So you can apply pico-Newtons on this molecule, and then you can stretch it. And then you can actually measure how much work you perform in these systems. And because it's a stochastic system, whenever you stretch it, it will perform a little bit. So you will need a little bit different work actually to do this. And if you measure this in an experiment, what you will see for these distributions, that you sometimes even have negative work. So this means you gain while you fall. And this is counterintuitive, but the idea is that sometimes you're lucky and you have a fluctuation that actually kicks you in the direction that you want to go. However, what is more important is that the averages still obey the second law of thermodynamics.
You mean when you pull, you add energy to the system?
Oh, you gain. You get from
Who
the
gains? The system? Or?
yeah, I mean, then it's the minus, yeah. How do you define the minus sign? But the idea, what I want to say here is that there are now new concepts where you define, for example, work, heat, and entropy on a single stochastic trajectory. And then you can also talk about for example, the distributions. However, now we are turning to to one of my favorite systems in biology, which is I want to introduce a little bit because I find it super remarkable. It's an active process that we have and also a lot of animals in our ears. And the ear is also interesting because it has been studied by many physicists. This is what people mostly don't know. For example, Helmholtz, famous Helmholtz, wrote a whole book on the sensation of sound and theory of music. And then there was also Ernst Mach and Max Planck. They had some experimental studies and also theoretical papers about attentional hearing, which is also very interesting because we as humans can also focus with our hearing. For example, if we later have drinks, I can talk to people, although the background is actually louder than the voice of the person that I talk to. And then Thomas Gord, also an astrophysicist, proposed in his PhD thesis that the ear must have some kind of active process because there's so much damping in that ear going on that it cannot resolve or cannot work how we know it. It's actually interesting, that was 1948, and then Berger got the Nobel Prize, talking about Nobel Prize, like 20 years later for the wrong theory, because he actually claimed that it's a non-active, purely mechanical process, because, yeah, interesting from a historical point of view. So if you look into an ear, very briefly, so if sound goes into the ear, you have the cochlea, and in the cochlea are these specialized cells, and these cells have a very beautiful structure. So here you see one. They have this very peculiar structure on the top, which is called a hair bundle, actually, because it has these hairs, these protrusions, and then a mechanical perturbation will displace these bundles, and then ion channels open, and this is how we hear. What is remarkable about our ear, it has a high amplification, so you can hear very, very faint sounds, actually. It is very well tuned to frequency, so even musicians, or some musicians are even better, so you can have a frequency resolution of 0.2%. It has a nonlinear compression. That means actually by definition that we can hear between 0 and 120 decibel, and decibel is a logarithmic scale. So if you compute the power, it's 10 to the 12 in power, actually, which is remarkable. And this is all, or this all contributes from an active process in the ear. And maybe some of you know this also, that our ears, they also send out sounds, very faint, but we lose it also when we get older. But babies, for example, also what they do here in the hospital, if you have a newborn, they actually measure the sound that is emitted by the ears, because if the baby emits sounds, the ears are usually healthy. And usually if they don't emit sounds, then there's some fluid usually in the canal.
It speaks through the ears.
Exactly. But it's also interesting because, I mean, I can talk forever about the ears, but I don't have much time. It's actually interesting that the spectrum, that every person can be identified because with their spectrum of the emissions. It's like a fingerprint.
Oh, wow.
Because it has probably to do with some defects in these membranes inside. Okay, so here what is also remarkable, that it's still some debate where this active process comes from, but we believe that the active process has to do with these hair cells, because if you isolate them, and it's a very complicated experiment, but what you can do here, you can image them from the top, and then you see actually that these hair cells, I hope you can see this here, you can appreciate that they start to wiggle. So it's a spontaneous movement, what they do. We can record these movements, and they look beautiful for physicists because nonlinear oscillations, relaxation oscillations here, for example, and then sometimes they also show these more bistable oscillations.
But it has for each person a dominant frequency.
For these, this is not the. This is actually now the movement of these hair cells.
Yeah.
Yeah, and these are actually from a frog, so this is not a human. That's how we cannot find humans. They will donate their ears for doing experiments that I can do some modeling on it. You know, that's ethically is still not accepted. So this is why we should have frogs. Yeah, why we use frogs here. So the first question one can ask is: are these? If you see something that moves, can you actually say, is this an equilibrium movement or non-equilibrium? Right, so this is a very naive, simple question. Right, so is this equilibrium or non-equilibrium? To determine this,
Well, if it's its eigenfrequency, then it's equilibrium, I would say.
I can also drive a pendulum with noise.
Yeah, but it looked like it's going to be.
It looks like, yeah, but we want to have a number, for example. Is it, how far is it away from equilibrium? Is it not? So one thing you
Did
can
you
do.
see the picture of this child on the swing?
Are you also saying that's spontaneous? I mean, it has a frequency.
Okay, but then yeah, we can yeah. Okay, so maybe we can discuss these details later. And I just want to say what I think is a clever idea here is to look at something which is called the fluctuation dissipation theorem. So the idea here is actually quite simple, that you say you have a system in a thermal equilibrium, for example, like here a Brownian particle, and then you will see some fluctuations. However, if you would perturb this particle with a very small force, then this perturbation will relax exactly the same as a fluctuation. And this is only true for equilibrium systems. So if we see that this fluctuation dissipation relation is broken, that is a good indication for a non-equilibrium system. And this is actually what you can often do here and measure.
But there is a non-equilibrium version of this. I mean, you can do it non-equilibrium. So
You can do it non-equilibrium, and
no,
then
I mean the fluctuation dissipation theorem. There is a non-equilibrium version, but this is just a special case. So
exactly.
So you should use the non-equilibrium fluctuation dissipation theorem, I would say, to check this. Right?
Yes, you can also do this, but here only for the statement to say if it's non-equilibrium or equilibrium, what you can do is you can use the equilibrium fluctuation dissipation theorem and show that it's violated.
Yeah, but yeah.
I think logically there's no problem with this, and this you can do. However, I come now to the next thing, because I think from the stochastic thermodynamics, there was also a very, it's not my result, from Harada Sasa, so what they found is actually that this violation, so here is a correlation function, and this is a response function. And in equilibrium, this here is 0, and in non-equilibrium, they can actually relate this with an equation to the average heat that is dissipated in the system. And this is remarkable, I think, because it's an equation. Of course, there are certain conditions for this. For example, one problem is it's actually a correlation in velocity. And so what I did, I transformed this into a correlation of position, because it's easier to measure position than a velocity, because a velocity is always a derived quantity. You also have to measure two things like position and time. So this is much easier to measure. However, it's also not easy to measure, because the problem here is it's an integral over in frequency space and Fourier space, so you have to measure all frequencies and from minus infinity to plus infinity, which is also technically very difficult. So it's impossible actually. So the idea here was to start with a linear model for these oscillations, and so it's a linear model of two equations. This is the most simple way to describe these oscillations, and from this I can derive now this violation function. And I think I'm also running maybe a little bit out of time here. So the idea is now we can fit this model to measurements, and then in the end we get a number out of this, which is then that this oscillations dissipates 146 kT per cycle. So I'm happy with this. Biologists would ask why is okay, maybe 500 or whatever. Why? And I think it's interesting because we can relate this directly to ATP, which is a molecule in the cell.
What's KT and what's ATP?
So kPt is the Boltzmann constant, and T is the room temperature of the experiment. And ATP is actually a molecule that is the energy budget in a cell. So from this, I know that this process needs roughly 60 ATPs per second. And with this, I have a lower bound. And now we can talk about certain mechanisms, which is, then we can talk about things going on.
This is per cell, 60 ATP per cell per second.
Per cycle.
Per cycle, right?
Yeah, or sorry, per second. So it's a lower bound.
How many of those cells are there in the year?
10,000 roughly.
It's not a lot of energy.
No, it's actually a lower bound. It's not a lot of energy. Okay, and then with this, I think I had a other part where we do now a mechanical model with a non-Markovian, but I will skip this. Sitting there, I will skip. I talk too much about what's happening in the beginning. Yeah, I just want to show something very briefly, which is the next step. Because I'm also very interested if someone of you has some experience in control theory. Because what we can do now, what my colleagues can do is actually we can activate certain molecules in space and time. So the idea here is it's called optogenetics. So here you see some algae, and then the light is coming from this side. So one of this species will go in this direction away from the light, and then these big ones, they go towards the light. So this has been now identified, and these molecules can be that are actually that causes this movement. They can be extracted, and now people can build now this complete zoo of molecules you can activate with light. So the idea is now, for example, in a cell here, this is a cell, and then you can switch on the light, and with the light you can activate now molecules. And you can do this globally, but you can also do this more locally by changing, for example, the light pattern here. I can show you here. So here you see the blue box. This is where we have a light pattern, and then the cell becomes active in this light pattern and starts to walk towards this direction. So what we did with this is the idea now to build smart microscopes. So a microscope will take an image, does image analysis, segmentation. Now we also started with deep learning here. And then we have a control algorithm where we say, for example, this should happen in the cell. And then the idea is that the optogenetic stimulation, so the pattern should be optimally chosen such that this target, what we want, is actually achieved. So here I show you, next I show you one example. For example, here we have a cell, so we can give it a path. And then this is completely automated now. So the microscope will look at the image, does image analysis, sees where the cell is, knows where the path is, and then it adjusts this blue light pattern such that the cell will walk on the circle. And this goes over 4 hours, but no one sits behind this microscope. So with this, we can now also direct cells. We can have several cells at the same time. However, what I want to end here is that the mathematics that are behind this control theory are very, very simple at the moment, but they're very robust. Yeah, we use more complicated things with Lyapunov stability analysis, and we also use Kalman filtering and all these kinds of things, but they did not work out because our systems are very noisy, and each cell is a unique person. So some like it, some don't like it. So if someone has more knowledge on control theory, I'm very happy to talk about this. And then with this, I would like to end the talk. And I still want to argue that what we are trying is we want to bring these 4 steps from physics to biology. I did not talk about our approaches for observing biological systems. There we use a lot of deep learning at the moment. Here also is an automated segmentation of the deep neural network that can color different samples. And we also use a lot of big simulation systems here for intracellular processes. Then what I think will be the future is here perturbing cells. Here it works actually on a square. Then this other one comes up here. I don't know what they do. Okay, so there.
Very much. Fascinating.
Yeah, it's next time I can also talk more about mathematics.
Can you also put them in a certain shape and arrange them in some pattern?
Yes. Do you mean the cells?
Yeah, the cells. Like organoid people, they're making arrangements in lung cells in a monotube or something.
Yeah, exactly. So we can also make mazes.
Yeah.
Yeah, because some surfaces, we can coat the surface with like different ligands and then it can only walk this way, or in channels, for example, that's possible. I mean, for the organoids, this is actually the long term, this is 2D, and 2D is simple. And 3D, if you have an organoid structure, then it's more complicated, but of course that would be an amazing experiment that we can rearrange cells in an organoid, because we can just say they should walk this way or that way, right?
Yeah, that would be fantastic.
That would be fantastic.
Applications to human beings.
I argue no, but that's of course my argument at the moment. In a human being,
So like therapy and psychiatry in the 19th century is not based on this.
no. I mean, if you want to know the current news about, because optogenetics has also been done in neurons, so you can activate neurons when you shine light on them.
Yeah.
And this is something that is now very promising, because you can use, I mean, you use animals and then you can activate certain brain regions and you can see how they behave. Yeah, but I don't know if you ever saw these experiments. I mean, you have this poor animal with an open skull, and then you have like these hundreds of optogenetics cables in this animal, and then you do behavioral experiments. But there's hope, because there are some animals, like birds, for example, they can sense magnetic fields. I don't know if you notice, they align to magnetic fields. And the hope is now to understand, instead of using optogenetics, to use magnetogenetics, right? Because then you don't need light, you need a magnetic field to activate molecules. However, this is something I think we will need another 50 years. But that would be for humans, of course. And
So what cell is that?
this is a good question. These are neurophils, I think. So very active cells. Because the student who there was a physicist, and he did not want to, we had also slower cells, but then you have to always, you know, then experiments take a long time, so he wanted to have like fast moving cells to do experiments.
But the idea is, as far as like an experimental toolkit, it works with human cells.
I think these are human cells. I mean, that's not
It's not for sure to
the problem is it does not work in humans.
right.
It works with human cells.
Yeah.
Yeah,
But if you got like some cancer growing in a petri dish, that
exactly.
Can you say something about the part you skipped?
I'm not sure whether that's a fair question.
The answer is yes.
Yeah, yes, I can say something, you know. Should I say something? It's the next question.
Over drinks, I suggest.
Yeah.
Okay.
Okay, good.
Okay, thank you very much.